Okay, guys, so in this lecture, we will be discussing about the cell cycle, its importance, and how the cell cycle it works. Cell cycle means that a cell is going to be replicated and going to be divided into two identical daughter cells. If we look into the phases of the cell cycle is a four-stage process consisting of gap 1, which we call G1. Then the stage of synthesis, S, gap 2, G2, and mitosis, M, which a cell undergoes as it grows and divides. After completing the cycle, it either starts the process once again from G1, or it may exit the cycle and goes into G0 or G0, where no further replication is going to take place. From G0, the cell can undergo into the terminal differentiation. The stages between one mitosis and the next, which include G1, S, and G2, they are known collectively as the interphase. Why this cell division is important? Cell division is a process of reproducing these cells, and it occurs during growth, during repair, and during the development of tissues. Well, if we look at the cell cycle, this is the cell cycle. And these are the repeating sequence of cellular growth and division throughout the life of an organism. G1, S phase, and G2. This is the longest phase, having 3 subphases, and this is known as interphase. Then comes the mitosis, prophase, metaphase, anaphase, telophase, and cytokinesis. The actual cell division occurs during this mitosis phase. Interphase, it is dependent, like we have just seen in the previous slide, that there are 3 subphases. First, growth phase, then DNA synthesis phase, and then the second growth phase, G1, G2. First growth phase, G1. Cell grows rapidly and carries out routine function. Phase takes most of, like this phase, it takes most of the cell's life. Muscle and nerve cells, they don't divide that frequently, or some of them, they don't even divide. So they remain in G1. Interphase. Now the synthesis phase. Here, cell's DNA is copied. At the end of this stage, each chromosome consists of 2 chromatids, which are attached at the centromere. Well, then the second growth phase starts, and here hollow microtubules are assembled. These microtubules, they are used to move the chromosomes in the center during the cell division, or the replication of the cell, or we can say when the cell is going to be divided into 2 daughter cells. Second phase of the cell cycle, G2. Now, after that, G2, there will be mitosis, interphase and mitosis. Mitosis, in this phase, this was G1, S, the synthesis, and then the G2. Over here, we have mitosis. Nucleus is divided into 2 nuclei. Each nucleus ends up with the same number of chromosomes as the original cell. This phase includes prophase, metaphase, anaphase, and telophase. Finally, there will be cytokinesis, and during this final stage, the cytoplasm, it divides equally between the 2 daughter cells. Interphase, as I told you, the first phase, the stages between one mitosis and the next mitosis, it will include G1, S, and G2, and they are known as interphase. M phase, mitosis followed by cytokinesis, cell separation, formation of two identical daughter cells. Now over here we can see prophase. Chromosomes, they condense and become visible. Spindle fibers emerge from the centrosome's nuclear envelope. It breaks down and the nucleolus, it disappears in prophase. Prometaphase, chromosomes continue to condense. Kinetochores, they appear at the centromeres. Mitotic spindle, microtubules, they attach to kinetochores, and centrosomes, they move towards the opposite poles. Metaphase. Mitotic spindle is fully developed. Centrosomes are at opposite poles of the cell. Centrosomes are lined up at the metaphase plate, and each sister chromatid is attached to a spindle fiber originating from opposite poles. In anaphase, coherent proteins binding the sister chromatids together break down, and sister chromatids, now we call those chromosomes. They are being pulled away towards the opposite poles. Non-kinetochore spindle fibers, they lengthen and they elongate the cell. Finally, it will be telophase. Chromosomes arrive at opposite poles and begin to condense. Nuclear envelope material surrounds each set of chromosomes. The mitotic spindle breaks down. Finally, there will be cytokinesis. And over here, the cytoplasm is going to be distributed equally between the two daughter cells. So here we are concerned with the animal cell at the moment. So a cleavage furrow separates the daughter cell. G0 phase or G0 phase. Some cells, they divide constantly while others are quiescent. They are not dividing further. These cells exit the G1 and enter a resting state, which we call G0. They don't go into the further division. In G0 or G0, a cell is performing its function without actively preparing to divide. So G0 or G0 is a permanent state for some cells, while others may restart division if they get the right signal. Regulation. The progression of cells through the cell cycle is regulated by various checkpoints at different stages. These checkpoints detect damaged DNA and prevent affected cells from replicating and dividing. The restriction point R is a crucial checkpoint located in the G1 phase. Most cells that pass through the R point will complete the entire cycle. Other checkpoints are located at the transitions between G1 and S and G2 and M phases. When damaged DNA is detected at any checkpoint, the checkpoint activates and increases the production of the p53 protein. p53 is a tumor suppressor gene, halts the cell cycle progression and initiates repair mechanisms for the damaged DNA. If the DNA cannot be repaired, then p53 ensures that the cell undergoes apoptosis, preventing it from replicating. Here we can see the checkpoint. Another checkpoint, another checkpoint. Regulation. In the context of oral cancer, several tumor suppressor proteins play crucial roles in preventing the development and progression of the disease. Notably, the p53 protein and retinoblastoma protein are key examples. p53 protein, we have discussed it a little bit in the previous slide. p53 is often referred to as the guardian of the genome. Due to its crucial role in maintaining DNA integrity in oral cancer, mutations in the TP53 gene, which encodes the p53 protein, are common. When functioning correctly, p53 halts the cell cycle in response to DNA damage, initiates DNA repair processes, and induces apoptosis if the damage is irreparable. However, mutations in p53 can disable these protective mechanisms, allowing cells with damaged DNA to proliferate, contributing to cancer development. Retinoblastoma protein, RB. The RB protein regulates cell cycle progression by controlling the transition from the G1 to the S phase. In oral cancer, inactivation of RB, either through direct mutations or through dysregulation of upstream regulators like CDK4 or 6, can lead to uncontrolled cell division. Phosphorylation of RB by cyclin-dependent kinase, CDKs, inactivates its tumor-suppressing function, thereby promoting the cell proliferation. In addition to p53 and RB, other tumor suppressor proteins and pathways are also implicated in oral cancer, like p16INK4A. The p16INK4A protein inhibits CDK4 and CDK6, preventing RB or retinoblastoma phosphorylation and thereby maintaining its ability to suppress the cell cycle progression. Loss or inactivation of p16INK4A is frequently observed in oral cancer, leading to unchecked cell division. Phosphatase and tensin homologue, PTEN. PTEN is another tumor suppressor that negatively regulates the PI3K AKT signaling pathway, which is involved in cell survival and proliferation. Mutations or loss of PTEN function can contribute to oral cancer by promoting cell growth and survival. The tumor suppressor proteins, through their roles in regulating the cell cycle, apoptosis, and DNA repair, are critical in preventing the initiation and progression of malignancies, including oral cancer. Understanding the functions and the consequences of their inactivation provides valuable insights into the molecular mechanisms underlying the disease and can guide the development of targeted therapies. Cyclin-dependent kinase, CDKs, which we were discussing. So these cyclin-dependent kinase, these are the family of protein kinase or enzymes that play a crucial role in regulating the cell cycle. They are key enzymes that, when combined with regulatory proteins called cyclins, control the progression of cells through the cell cycle's various phases. Here we can see just an example how the CDKs, different CDKs, they work and at which stage. Key functions and roles of CDK. Regulation of cell cycle progression. CDKs drive the cell through different phases of the cell cycle by phosphorylating target proteins. This phosphorylation triggers specific events required for cell cycle progression, such as DNA replication and mitosis. Activation by cyclins. CDKs are inactive in their monomeric form and require binding to cyclins to become active. The levels of cyclins fluctuate throughout the cell cycle, ensuring that CDK activity is tightly regulated. Phases of cell cycle and CDK. Different CDK cyclin complexes operate at specific points in the cell cycle. G1 phase CDK4 and CDK6, when bound to cyclin D, help the cell progress from G1 phase to the S phase. S phase CDK2 paired with cyclin E initiates DNA replication. And G2 and M transition, CDK1 in combination with cyclin B drives the cell into mitosis. Checkpoint control. CDKs are involved in cell cycle checkpoints, ensuring that cells do not proceed to the next phase until certain conditions are met, such as complete DNA replication or repair of DNA damage. Clinical significance of CDK. Cancer. Dysregulation of CDK activity is a common feature in many cancers. Overactive CDKs can lead to uncontrolled cell proliferation. For example, mutations to overexpression of cyclins, loss of CDK inhibitors, or mutations in the CDKs themselves can contribute to oncogenesis, cancer production. Therapeutic targets, due to their critical role in cell cycle regulation, CDKs are targeted by cancer therapy. CDK inhibitors such as palbociclib inhibits CDK4 and 6 are used in the treatment of certain cancers to halt the cell proliferation. Certain examples of these CDK inhibitors: palbociclib, then ribociclib, and abemaciclib. These are the specific inhibitors of CDK4 and CDK6 used in the treatment of breast cancer. They work by preventing the phosphorylation of the retinoblastoma protein, thereby blocking the transition from the G1 to the S phase. Regulation mechanism: CDK inhibitors, proteins such as P21, P27, and P16 bind to and inhibit CDKs, providing an additional layer of regulation. Phosphorylation: CDKs are also regulated by phosphorylation and dephosphorylation of specific residues. For instance, the phosphorylation of CDK1 on tyrosine residue, which is TYR15, inactivates it, and dephosphorylation activates it. So this is inactivating and this is activating. CDK inhibitors. Examples of CDK inhibitors: palbociclib, ribociclib, and abemaciclib. These are the specific inhibitors of these enzymes. So we have discussed about it. And then we have discussed the phosphorylation. Research and future directions. Ongoing research aims to better understanding the role of CDKs in cell biology and develop more effective CDK inhibitors with fewer side effects for cancer treatment. Additionally, exploring the role of CDKs in other diseases such as neurodegenerative disorders in a growing area of interest. So in summary, we can say cycle-independent kinases are vital regulators of the cell cycle, and their precise control is essential for normal cell function and prevention of diseases like some cancers. So over here, we are done with this lecture.